Hello, everyone. I'm Mark Avigan. I'm an Associate Director in CDER's Office of Pharmacovigilance and Epidemiology at the FDA. As the session chair, it gives me great pleasure to welcome you. We will hear a series of rapid-fire talks on a comprehensive state-of-the-art consensus report for drug-induced liver injury, or DILI, that is just hot off the press. Next slide. Next slide. Now, regards our presentations and the report itself, the views that will be discussed are those of the presenters and not an official position of the FDA or any other regulatory organization. Next slide. The new report on DILI is the culmination of hard work by 40 extraordinary individuals from across the globe who met regularly for the last 3 years, a group of folks in the drug development community, regulatory scientists, and academic clinicians. And I was privileged to be one of the contributors in this enormous effort. We were convened to develop a wide-ranging consensus document as individuals with recognized but diverse expertise in the DILI space under the auspices of the Council for International Organizations of Medical Sciences, or CIOMS, an organization that brings together public health and health research stakeholders from across the globe on important issues. Next slide. The scope of the new DILI document is ambitious. It lays out a detailed framework and emerging tools to detect, diagnose, and manage DILI during drug development and in the post-market setting. With excellent support in our editing phase from Monica Zweigart at CIOMS, the entire DILI report is now available online for all of you to download. The link for the report is on the CIOMS website and is shown on this slide. Next slide. With the brief time that we have for this session, four of my terrific colleagues will discuss some of the highlights in this comprehensive document. They're Hervé Lelouet, who is the current president of CIOMS. Hervé is an academic hepatologist. He, with his colleague Lembit Rago, the Secretary General of CIOMS, conceived of the DILI topic for a CIOMS work group and gave us our charge. Hervé will very briefly discuss the goals, initiatives, and context of the 2020 report. We'll then hear from representatives or leaders of the three main sub-working groups that we established early on during the crafting of this report. They are Ari Regev, who is chair of the GI and Liver Safety Committee at Eli Lilly. Ari will discuss DILI detection, characterization, and assessment in clinical trials. And Ari will be followed by Raul Andrade, who's an expert in the field of DILI and a professor of medicine at the University of Malaga in Spain. Raul will discuss the identification and utilization of liver safety biomarkers and hepatotoxicity linked to the herbal products. And our last speaker, who has pre-recorded his talk because of a schedule conflict, is Walter Strauss, who's an associate vice president and therapeutic area head for vaccines and infectious diseases at Merck Research Labs. Walter will discuss DILI risk assessment, minimization, communication, and post-market. At the end of the session, we'll have a panel discussion. We'll be joined by Einar Bjornsson, who's chief of internal medicine at Landspítala University Hospital in Iceland, to answer questions that you might have using the Zoom chat box on your screen. So Hervé, with that, take it away.
Thank you very much, Mark. Good evening, good morning, hello everyone. I'm Hervé, the president of CIOMS, and I would like to thank very much, first of all my colleagues for the wonderful job they did. It was a really wonderful group, and it was not so easy at the beginning, but they really worked a lot. The commitment were full, and it was a wonderful experience. I would like to thank also the people from the colleagues from the Duke-NUS University and the Center of Regulatory Excellence for organizing this session. I would like, in the next few minutes, it will be very brief, don't worry, to present you what is CIOMS, what it does, and what was the context of the DILI initiative. Next slide, please. So the Council for International Organization of Medical Sciences has been founded in 1949 by the WHO and the UNESCO. It is currently in official relation with WHO and ICH observer since 2016. The mission is to advance public health through guidance on health research, ethics, medical products development, and safety. Next, please. So, in short, the organization is located in Geneva. It's international. it's non-governmental, it's not for profit, and it's in official relation with WHO plus associate partner of UNESCO. So for WHO and other members like health authorities, academic organizations, pharmaceutical other stakeholders, it is an organization of medical sciences organization. And finally, we are a forum for discussion and neutral platform to elaborate new ideas in drug development, pharmacovigilance, and research and ethics. Next, please. So the working groups are the core of CIOMS. And currently we have a working group on MEDRA implementation, clinical research, patient involvement in development and safer use, benefit-risk balance for medical products, real-world data and real-world evidence in regulatory decision, and of course, the working group on drug-induced liver injury. So next, please. So what was the situation before? And without all the colleagues, really, it was an effort. And they all did it because at the beginning it was not so obvious that we were able to create this type of group. So there were several initiatives from academics, regulatory, industry, and patient organization that needed to be coordinated to avoid redundancy, improve dissemination, and widen the audience. So it is because all the collaborators were clever enough to understand that we need to go ahead that they created the CIOMS DILI Working Group. And once again, I would like to pay tribute to all my colleagues from this group because it was a fantastic experience. The next, please. So it's now time to give the floor to Ari.
Good morning, good afternoon, and good evening, everybody. I hope you can see my screen. I will present today on principles of detection, characterization, and risk assessment of DILI in clinical trials. These are three major topics in the field of DILI, which are thoroughly discussed in the CIOMS consensus paper. Unfortunately, it is impossible to cover these three topics in 15 minutes, so I chose a few points, which I will try to highlight in the next few slides. I encourage everyone to look at the final summary of the working group for additional information. So this is the outline of my talk. I will start with a few basic concepts related to DILI and related to DILI classification. I will touch on some of the DILI phenotypes, including the patients with pre-existing liver disease. I will then talk briefly about principles of DILI detection and risk assessment during drug development. Next. So a few words on general classification of DILI. Next. This table is based on a review paper published in the New England Journal of Medicine last year. This is one of 527 publications cited in the CIOMS consensus paper. As you can see, drug-induced liver injury is divided into three major types: direct, indirect, and idiosyncratic. So direct DILI is caused by agents that are intrinsically toxic to the liver. This type of liver injury is relatively common, predictable, dose-dependent, and often reproducible in animal models. The time to onset, or what we call latency period, is typically short, usually between 1 to 5 days after ingestion of a high dose. The classical example of this subtype is acetaminophen overdose. Other examples include niacin, cocaine, and cancer chemotherapy. Idiosyncratic hepatotoxicity is caused by agents that have little or no intrinsic toxicity. These agents cause liver injury only in rare causes, typically 1 in 2,000 to 1 in 100,000 exposures. This type of liver injury is typically unpredictable, not dose dependent, and not reproducible in animal models. The mechanism is linked to a yet incompletely understood combination of the drug's attributes and individual host susceptibility. There are many examples of drugs causing this type of DILI, which is the most concerning for healthcare providers and for drug developers. This is the type that leads to most of the early terminations of drugs during development and most of the post-marketing limitations on drug use. Indirect hepatotoxicity is a relatively new category. It is largely the result of the increasing numbers of new drugs and a broader view of the definition of DILI. Indirect injury can represent induction of a new liver condition or an exacerbation of a pre-existing condition, such as induction of immune-mediated hepatitis or worsening of hepatitis B or C or fatty liver disease. The frequency of indirect hepatotoxicity varies, but it is typically higher than idiosyncratic DILI, although lower than direct DILI. The classical example is immune-mediated liver injury caused by checkpoint inhibitors, on which you will hear more in the following presentations. The three types of DILI can be manifested by distinctly different patterns of clinical features or phenotypes. It is important to remember that DILI may mimic almost any known type of liver injury. Next. And this slide shows a few of the known DILI phenotypes. Acute hepatitis resembling viral hepatitis, which is here first on the list, is the most commonly encountered phenotype of idiosyncratic DILI. It can be caused by many drugs, and it causes most of our concerns during drug development. One of the typical examples is isoniazid-related liver injury. However, it is increasingly recognized that other forms of DILI can occur during drug development and can be serious and even life-threatening. Cholestatic and mixed liver injury, which is the second on this list, are probably second on our list in terms of incidence. The most common example is amoxicillin clavulanate, also known as augmentin. Acute hepatic necrosis, which is the third on this list, is another relatively common type, and it is most commonly related to acetaminophen. Although other types are less common, they are associated with well-known drugs, and they are potentially lethal. I'll be happy to elaborate on these types during the Q&A session. Again, it is important to remember that DILI may come in different shapes and forms. It does not necessarily have to present with high ALT or AST values. And even in the absence of high ALT or AST, it may lead to serious outcome. And again, the type of DILI that we encounter most commonly during drug development is the acute hepatitis phenotype. The exact case definition of DILI is still a matter of some discussion. The most commonly used definition has been proposed by an international DILI expert working group in 2011. According to this definition, any one of the following criteria is indicative of DILI once other causes of liver injury have been systematically excluded. ALT equal to or higher than 5 times the upper limit of normal, ALT equal to or higher than 3 times the upper limit of normal, and concurrent elevation of total bilirubin exceeding 2 times the upper limit of normal, or alkaline phosphatase equal to or higher than 2 times upper limit of normal when the source of increased alkaline phosphatase level is the liver. Importantly, according to this DILI expert working group, isolated hyperbilirubinemia is usually not DILI, and isolated elevation of GGT is also insufficient to qualify as DILI. However, these criteria may not be applicable to patients who have pre-existing chronic liver disease. Detection and assessment of DILI in patients with pre-existing liver injury is potentially more complex and often a cause of confusion. Presently, a substantial proportion of clinical trials enroll patients with pre-existing liver disease such as NASH, viral hepatitis, alcohol-related liver disease, and metastatic liver disease. DILI phenotypes may be considerably different when superimposed on an underlying liver disease, especially if there is a severe pre-existing hepatic damage with hepatic impairment. DILI may be difficult to differentiate from a fluctuation in pre-existing disease. Detection may be delayed since ALT and AST may be only mildly elevated or may remain within normal range. Outcome may be more severe, including a higher mortality. And finally, it is important to remember that pre-existing hepatic impairment may lead to increased exposure and a higher risk of dose-dependent hepatotoxicity. I will now turn to detection and risk assessment of DILI in drug development. First, a few words on preclinical assessment of DILI risk. Idiosyncratic DILI, and to a large degree, indirect DILI, which are the main concern of drug developers, are difficult to predict using non-clinical methods. The ability of various non-clinical assays or studies to predict the risk of idiosyncratic and indirect DILI is still a matter of intense debate. Assessment of reactive metabolite formation, mitochondrial toxicity, animal toxicity, studies and various modeling approaches have both strong supporters and severe critics. There are studies that find these methods to be of high predictive value, while others find them to be of low or no predictive value. So how do we assess the risk of idiosyncratic hepatocellular DILI during drug development? It still looks like the best approach to detecting DILI and assessing the risk of severe DILI is based on a comprehensive and careful hepatic safety data collection and assessment during clinical trials. Cases of mild and moderate liver injury are often the first signal of a DILI risk. Understanding the difference between benign, quote-unquote, ALT elevation and a potential DILI signal is critically important. This depends on effective monitoring and differentiation between what we call adaptation and actual liver injury. It also depends on analysis of extreme values of liver tests or outliers rather than mean or median values. Additional important tools in assessment of DILI risk are the identification of Hy's law cases and correct use of the Yiddish plot, as well as effective causality assessment to differentiate between DILI and liver injury due to other causes. Lastly, there is a strong connection between our ability to understand the hepatic safety profile of a drug in development and adherence to predetermined hepatic stopping rules. In summary, DILI is divided into direct, idiosyncratic, and indirect hepatotoxicity. As novel drugs are being developed, new DILI phenotypes are encountered and should be kept in mind and looked for. DILI may present with various phenotypes, and these phenotypes may change in patients with pre-existing liver disease. In such patients, severe liver injury may occur without ALT or AST elevation, which requires careful monitoring and increased awareness. The value of non-clinical methods for the prediction of idiosyncratic and indirect DILI is still controversial. The most useful approach for DILI detection and risk assessment is based on comprehensive hepatic data collection, assessment, and interpretation during clinical phases of drug development. And my last slide, I would like to thank again the entire CIOMS working group for the hard work and perseverance. And special thanks to Hervé Lelot, the president of CIOMS, who is here with us, and Rago Lembit, the Secretary General of CIOMS, for their great leadership of this very important project. Thank you very much.
Thank you. Good evening, everyone. It's a pleasure for me to join this important group, CIOMS group, and also to participate in this webinar. My task is addressing liver safety biomarkers. It's a chapter of this, the CIOMS documents, and also a brief comments on herbal and dietary supplement hepatotoxicity. Next slide, please. According to the US FDA, a biomarker is defined as a defined characteristics that is measured as an indicator of biological processor, pathogenic processor, or biological response to an exposure or intervention, including therapeutic intervention. But it's also important to understand biomarkers is the context of use as is described in this slide, the context of use for the FDA and for the EMA. And next slide, please. For example, the context of use can be diagnostic biomarker. This is the case, for example, of ACV RNA, which is used for the diagnosis of active hepatitis C, but also this biomarker can be a biomarker of efficacy pharmacodynamics. For example, in the case of use of drugs for hepatitis C, the ICV RNA clearance is a biomarker of efficacy, as is the normalization of ALT. There are also biomarkers for monitoring safety. For example, is the case of ALT, measured at baseline and during therapy with the drugs in clinical trials. Also, there are biomarkers that are predictive of a given event. For example, biomarkers that are measured, for example, genetic biomarkers in individuals who develop the adverse reaction against individuals who do not develop the adverse reaction. This is, for example, the case of HLA-B5701 in the case of flucloxacillin hepatotoxicity or abacavir serious adverse reaction. And there are also prognostic biomarkers, which predicts the likelihood of a future event or disease progression. This is the case, for example, for MEL score. It's important also to remind that there are currently no liver safety biomarkers qualified by regulatory authorities. In drug development and for detection of DILI, serum ALT is used as a biomarker of safety, and total bilirubin and INR are biomarkers that measure hepatic function, severity of the potential liver damage. And for the discovery of new biomarkers, it's important to take into account the sensitivity, specificity, the positive predictive value, and the negative predictive value, and finally the accuracy of the biomarker, the area under the cure, in comparison with established or traditional biomarkers. For example, ALT is used as a biomarker for liver injury, but is really not specific for liver injury. And total bilirubin is used as a biomarker which is sensitive for severity, and MELT is used as a accuracy biomarker for prognosis. So for this process of discovery of new biomarkers discovery is important to have a biobank, and for all subjects, including clinical trials, with the medication of interest and the placebo, to have baseline and serial samples. And for qualification, it's important to validate biomarkers in independent data sets. It's important because it is a major issue in new biomarkers, is the standardization of the values of the new biomarker, precision, accuracy, reproducibility. This table showed a list of biomarkers which are under discovery and validation. You can see that some of them, for example, total cytokeratin 18, have received a letter of support from the FDA and the EMA. These biomarkers are really mechanistic biomarkers, have shown many of them to be elevated earlier during the cures of DILI, earlier than traditional biomarker like ALT. The two last in the table, the macrophage colony stimulating factor receptor 1 and the osteopontin are biomarkers for prognosis. For example, you can see in this slide that for a series of 15 patients who died out of 133 from the DILI network, you can see that the performance of these two prognostic biomarkers ranked, for example, immediately after the INR. The INR was the more accurate biomarker for predicting prognosis. This is a traditional biomarker, but the osteopontin ranked second. For example, you can see 0.86, and followed by the total bilirubin. And this, in the bottom of the slide, you can see that there is also a study from a group, the Indiana group, which is a DILI group, that in their cohort, they assess the influence of comorbidity with the Charlson comorbidity index, and in the logistic regression model, they found that also MELT and albumin were independent variables associated with 6-month mortality after DILI onset, and they were able to make a calculator with these 3 variables, the Charlson comorbidity index above 2, plus MELT plus albumin, with a very good accuracy, an area under the curve of 0.89. An important barrier with predictive DILI biomarkers is that due to the low incidence of this adverse reaction, drug-induced liver injury, the positive predictive value may be low. And in addition, for DILI, and also for other adverse events, there is a need for a clear case definition, exclusion of other causes, a well definition of the phenotype and pattern, and there is currently no gold standard for that. So it's important to collect biosamples from all patients, including in a clinical trial or in a prospective study, to take care really in specimen collection, handling, and processing. This is the example that I commented before for the case of flucloxacillin. In a genome-wide association study, the HLA-B, class 1 HLA-B 5701, the same allele that predicts abacavir serious adverse reaction, was overrepresented in DILI cases. 80% of DILI cases have this allele versus 13% in the control population, with yield and odds ratio of 36. So this allele have a high negative predictive value, but the low positive predictive value was really the positive predictive value, was really low. And this is because this is a very rare adverse event. So this allele is present in around 7% of the population. So, this adverse reaction occurred very infrequently. So it was calculated that you need to test 1,000 patients to prevent one flucloxacillin DILI case. So this is not really useful like a preemptive biomarker, but because of the high negative predictive value, it may be useful for confirmatory diagnosis tests. Next slide, please. There are other biomarkers that were qualified, but they didn't perform quite well, such as bile acids, GSTA, and FADP. There are also other diagnostic causality biomarkers, such as the test of lymphocyte transformation or monocyte transformation, hepatocyte-like test, which is called meta-HEPS. The drug protein adults, which is not standardized in clinical practice. Also, there are diagnostic and prognostic imaging biomarkers, ultrasonography or MRI or elastography. As commented before, the predictive susceptibility genetic biomarker, which are really limited in DILI because of the rarity of the adverse reaction. And here I start with some comments about the herbal and dietary supplement hepatotoxicity. Herbal and dietary supplements are used to enrich diets and improve health function. This is the kind of use in general population, and they encompass several agents such as vitamins, mineral, amino acid, powder, herbals, botanical, etc. And the problem here is that they are regulated as foods, so no efficacy or safety testing is required. And another important issue is the variable manufacturing, which determines that there is a wide variation in batching. But the issue of herbal and dietary supplement use is really growing, and accordingly also the reporting of liver injury associated with these products. And in the right part of the slide, you can see data from in the top data from the DILI group, which this bar showed that there was an increasing in the reporting of cases across the years of registry working, and also in our registry in the bottom of the slide, you can see this bar that during the last years of reporting, before this paper was published, you can see that herbal and dietary supplements in general, but particularly anabolic steroids, androgenic steroids, experience higher reporting with a higher prevalence with regard to the conventional medication. So we have a serious problem here, and this is because of the generalized misconception that all natural is safer and more of the natural is better. And for example, these are data from the DILI Group published in Hepatology. You can see that for bodybuilding products, anabolic steroids, you can see that there was a high frequency of hospitalization. All cases were male, and in contrast for non-bodybuilding products, these products are really used in general for losing weight. Is a common indication of these products. You can see that there were more females and there were more severe outcome with more liver transplant and deaths. Actually, there was no liver transplant, there were no deaths for patients with anabolic steroid hepatotoxicity. And these are results that are comparable with results from our group. And I think this is the last slide. This is an interesting paper also published recently by the DILI Group that they try to analyze the ingredients of the herbal products, herbal and dietary supplements, and you can see that there was an inaccurate labels. This is a really big issue. Inaccurate labels for many of these products. So there was the label didn't match with the composition of the products. For bodybuilding, 80%, for weight loss products, 72%, etc. Next. So I think this is the last slide. Another important issue with herbal dietary supplements is the causality assessment. Is more difficult even than for drugs, for conventional drugs, and sometimes it's difficult to establish the precise latency, the challenge. It's important to pay attention to prior reports of phenotype. There are some information in databases such as liver tox for at least 50 HDS products. It's important to exclude, of course, other causes. And I think prospective DILI registry are helping in this. Ingredient analysis I think is an important addition. And the expert opinion used in prospective DILI registry is really more accurate for causality assessment than in general practice. And thank you very much for your attention.
Good evening. My name is Walter Strauss, and I will be presenting on behalf of our working group team best practices in post-marketing DILI risk assessment, risk minimization, and risk communication. Key background that you should be aware of. That firstly, we face a number of challenges. One challenge is that in the clinical trial setting, DILI is typically not identified because it's rare, and therefore clinical trials are usually underpowered to detect DILI cases prior to licensure. Consequently, most DILI cases are identified post-licensure. But there are several challenges there as well. Firstly, most clinicians are not terribly familiar with DILI. and the clinical practice setting is not generally attuned to identify DILI. For example, patients are monitored less intensively than they are in clinical research. And most providers outside of hepatologists are not as familiar with identifying hepatotoxicity, particularly early signs of hepatotoxicity. There's also a lack of a deep familiarity with the product labels that contain information about the overall product characteristics, as well as a general lack of familiarity with relevant clinical practice guidelines. And I take isoniazid as an example of that. In order to develop recommendations and to assess the situation, the working group considered key international pharmacovigilance documents, such as documents developed by CIOMS itself, the Working Group 8 Practical Aspects of Signal Detection and Pharmacovigilance, and the ICH Technical Requirements for Pharmaceuticals for Human Use, E2E guideline on planning pharmacovigilance studies. We reviewed those documents, and our recommendations build upon those, as well as incorporating recent developments outside of those documents that are pertinent to DILI. The current post-marketing surveillance arena can be broadly divided into passive surveillance and active surveillance categories. And overall, the intent of the post-marketing surveillance effort is to identify susceptibility factors and signal detection for idiosyncratic DILI in the general population. But there are some challenges with the current status. Passive surveillance refers to the voluntary spontaneous reporting systems that you're familiar with. These involve the expectation that providers in particular will identify and report cases of DILI that come to their attention. These systems are in place in much of the world, but they're limited throughout the world by underreporting. There's a recognition that higher adverse event reporting rates occur in those countries that happen to have mandatory reporting requirements, as you might expect. However, the reports that are received are often incomplete and missing key data elements needed to really inform causality assessment. For example, there may be missing data on concomitant medications and comorbidities. Active surveillance, in contrast, refers to prospective monitoring, typically involving a standardized data collection instrument, such as one that is used in a formal pharmacoepidemiologic study. There's also increasing interest in prospective use of electronic data sources, such as medical records and administrative or claims data, in order to identify novel risk factors and features of DILI, such as drug-drug interactions and time to onset. And as well, there is a tremendous amount of work being conducted on new technologies such as data mining and natural language processing, which I will not be discussing in detail. Key recommendations that are team developed for risk assessment are firstly a strong recommendation for signal detection of potentially hepatotoxic drugs, as well as herbal and dietary supplements that are in the marketplace, using such sources as the FDA's adverse event reporting system that receives these spontaneous reports, known as FAERS, the WHO's VigiBase, which is coordinated out of Uppsala, Sweden, as well as administrative or claims data that utilize ICD-10 codes. And that is true for much of Sentinel, which is a major FDA initiative on post-licensure safety assessment. It was also recommended to collect at least a minimum common set of data elements for all DILI case series publications. And the reason for this is that if there is a common data set, it facilitates inter-study comparisons and facilitates aggregating data. Those drugs that have validated hepatotoxicity safety signals that arise from spontaneous data sets should generally trigger an in-depth investigation In other words, within the WHO's VIGI base, if there is a safety signal that's identified, that should generally trigger a formal analysis, including comparative population-based studies to characterize and quantify the post-market risk of DILI. The working group was particularly interested in registries, DILI registries. And a registry for our purposes is an organized set of prospectively and systematically collected observational records, records of individual cases that incorporate a common minimum data set for the reasons I've just described on specified outcomes within a population that is defined by a particular disease, a condition, or exposure. And these registries are of particular value when one is collecting data on rare events. Generally speaking, large data sets, administrative data sets are not terribly useful for this sort of purpose, but they're ideal for collecting data on rare events. These registries can augment case detection and enhance signal detection in the general population. Furthermore, biological specimens that are collected through these registries can also be used to develop and support the development of new diagnostic and prognostic biomarkers. Here is a list of a number of DILI registries that were in place at the time of publication of the CIOMS report. In columns are the different registries. In rows are different characteristics of those registries, including when they were initiated, what sort of causality assessment tool they use, how many cases were enrolled at the time of the report, and whether biological specimens are available through these registries. And I will call your attention in particular for the purposes of this presentation to the Japanese DILI registry and to the National China DILI registry, applicable to obviously East Asia. These registries, the oldest ones were initiated in the early 1990s. Fortunately, almost all of them use the RUCOM instrument for causality assessment. They enroll anywhere from several dozen up to well over 5,000 records, and all of them collect biological specimens. Let me turn now to risk minimization and communication and talk a bit about the principles of risk minimization communication. Firstly, risk management is required. It's fundamental to mitigate the risk of DILI in real-world settings. And it refers to a structured and ongoing systematic approach to calibrate the evolving safety and effectiveness profile of a molecule, which is supported by actions intended to minimize risk of harm and to maximize benefit. The current focus of risk management is upon proactive signal detection. In other words, rather than reactive, proactive signal detection, as well as periodic risk evaluation. Fundamental to risk minimization and communication is the use of the product label. This is the core document that's employed by healthcare providers, intended for healthcare providers on product use. It summarizes the product characteristics and has legal standing in order to support proper dissemination of the product information so that accurate information about the product is provided to healthcare providers. I also want to tease out a couple of other terms that are used within risk management. One of these is risk stratification. And this is a tool that is used to categorize risk by segmenting a population that may be prescribed a drug according to expected likelihood of harm or benefit. So for example, age may be a risk stratifying characteristic, or renal function may be a characteristic of used to stratify populations. Currently available regulatory recommendations to detect and mitigate the risk of DILI have been developed within the setting of closely monitored clinical studies for the purpose of licensure. They're also used in product labeling, but we recognize that clinical trial populations are often relatively homogeneous, and the populations that ultimately use a product are going to be heterogeneous, and they, when the product is used in the so-called real world following licensure. Risk mitigation is a effort intended to further limit the risk of DILI in special patient populations, such as those with pre-existing liver disease or malignancies. This is an area of significant international interest, but it is one that we admit is a work in progress. Additionally, the information that's provided in product labels of drugs that are marked by similar risk often varies even within drugs within the same class as a result of many factors, and I'll discuss that in just a bit. So to that end, several challenges in effective communication of DILI risk in product labeling is firstly that there's confusion between liver function and liver injury. For example, the severity of liver injury and the degree of impaired liver function are different concepts, but they're often conflated or they're often confused with one another. For example, serum levels of aminotransferases measure cellular damage, but they're often erroneously considered to measure liver function, a different concept. An additional issue is reliance upon safety terms that are used in clinical trials, which is a very formal environment, different than the more heterogeneous world of clinical practice. For example, terms may be used imprecisely. Examples of these include autoimmune hepatitis and steatohepatitis. And then finally, there are also international differences in the use of certain liver failure terms, such as acute and subacute, acute on chronic, and chronic liver failure. Additional challenges in effective communication in product labeling are that there are differences across national regulatory authorities in terms of how product labels are the contents of product labels. The labeling practices vary by country, and this may lead to variability not only across countries, but even within the same drug class. Countries differ in terms of how they assess a product's benefit risk profile. They differ in terms of how of the structure and practices of their regulatory review divisions and the requirements of those review divisions. They're also differ in terms of their relative enthusiasm for rapidly adopting new scientific evidence. And we also have to be cognizant that medical treatments, practices, and guidelines differ by country and by regions of the world. Added to that, companies themselves often have some latitude to propose their own descriptions, their own nuanced descriptions in order to most effectively communicate risk and management. As an example of this, and I won't go through this in detail, but it's an example of differences within the EU, the European Union, summary of product characteristics as of November 19, and the US label. So on the left-hand column is the European label, on the right-hand column, the US label. And what you will see is that there is a different manner of describing impaired liver function and how to handle it within the warnings and precautions section of the European versus the US labels, with the European label saying patients should be monitored as appropriate for impaired liver function and be instructed to contact their physician in case signs and symptoms suggestive of liver injury occur, such as jaundice and vomiting. And conversely, on the US side, there is a narrower reference. In some patients, liver injury recurred upon re-challenge, providing evidence that this drug caused the injury. And then there is also a difference in terms of whether or not data from clinical trial are disclosed in the respective labels. So Optimal risk management for DILI requires, firstly, that risk factors are well characterized, that the known features of the liver injury due to a drug, that is the drug's signature, are well described. Risk monitoring and management need to be based upon reliable measures that can predict the outcome, highly reliable biomarkers, for example. The pattern and the course of the injury need to be well defined, accounting for possible variability that relate to different features of different patient groups. So in actual practice, characterizing DILI risk can be difficult due to some of these inconsistencies in nomenclature that I've mentioned, some uncertainties of the pathogenesis of DILI, the relatively limited data, the rareness of these DILI events, and low event rates. Management of DILI would be improved if there were a greater adherence to systemized data collection to characterize DILI, such as the use of registries that have been mentioned, additional research to characterize pathogenesis, as other speakers have mentioned, and then harmonization of reporting and labeling approaches across the world. Our recommendations on risk management and communication are, firstly, that the descriptions of product labels of risk for DILI, such as time course, the clinical pattern of disease, the mechanism of liver injury, as well as the recommendations for liver monitoring, should be informed by available product-related data. We recognize the role of peer-reviewed publications, which provide a rich source of developing information on the risk for DILI that's associated with a suspect marketed drug or a class of drugs. We also point the audience to regulatory agency websites and to the liver tox database maintained by the NIH, which provides useful additional information for clinicians to guide them in managing hepatotoxicity risk. And finally, the role of medical professional societies and practice guidelines is important as they provide valuable recommendations on the optimal use of potentially hepatotoxic drugs in patients. On behalf of the working group, I'd like to thank you, and I would note our team members, in particular Einar Bjornsson, who is our chair at the National University of Iceland, as well as colleagues from Michelle Bordolino, Stuart Geary, Uzo Shinobu, James Southern, myself, Xiaobo Wang, and Hui Talia Zhang. Thank you very much.
Well, thank you to all our speakers for a terrific summary of the CIOMS 2020 DILI report, which is really a big document and it really warrants having a look at. A lot of appendices, a lot of information that we couldn't really get into in too much detail. So it covers a lot of territory and has sparked a lot of interest. Now let's move on to our panel discussion, and Einar Bjornsson will kindly join us. Please use the chat box to share your questions, and I'll try to group them as best I can for our panelists to get in as many as possible. So to start things off, this is to bring Einar into the discussion. What is the role of the post-market pharmacovigilance and DILI risk assessment for a newly marketed drug or biological agent when there's already early information about its hepatotoxicity from clinical trials, where we already see a signal in clinical trials? What is the value of following these drugs into the post-market for further study? Einar.
Jean-Marc, I could just begin the answer of this question because I was a member of the Pharmacovigilance Risk Assessment Committee.
Okay, sure. So that's great. So Hervé, the question is that it's about the life cycle of a drug where we see in the clinical trial phase, we see a liver signal. What is the value of following the drug into the post-market and doing further pharmacovigilance and other post-market studies?
My opinion is that, like an expert from independent from the European Commission, is that finally it was very uncomfortable to make a decision about DILI because we had not any rules, any guidelines. And it was one of the reasons why I really, as a president of CIOMS, pushed to have this type of guideline. I don't know that it is the best, but at least it is something, and I think we must use it. But at the time when I spoke, it was to say finally, oh yes, we will follow during 2 weeks and 3 weeks, but without any argument, it was not evidence-based. So I think that at least this type of guideline will allow to have a more evidence-based decision, which is not so easy finally. So I don't know if
that's great. That's an important point. And I just want, and Einar, do you, again, from your point of view, you've done many studies in the post-market setting of drugs in particular with a liability for hepatotoxicity. From your point of view, what is the value of these post-market studies?
But probably Einar will answer.
Yes, I think they are of crucial importance because if there has been a minor signal, for example, with a drug that has very strong indication and an important drug, otherwise important drug, both the regulators should I think inform the companies that they should have a very close look at these signals that are appearing, and we have previously relied on spontaneous reporting, but I think we have to do better than that, and the companies should be responsible for collecting these signals, and also those, for example, specialties that are mostly involved in prescribing these new drugs, that they should have a responsibility of informing the authorities.
Well, let me just jump in there, because you're in Iceland and you have the Icelandic registry where you have a handle on everybody in Iceland with their medical charts and so on. So just if you could just for a moment just leave the spontaneous reports behind and just tell us a little bit about how you can use these tools in a system where you have such access to universal patient data.
Of course, maybe it's easier in a smaller country, but because we have a limited number of specialists that prescribes these medication. For example, we have detected a signal post-marketing with a new cancer drug that has been not. We've detected two clinically important reactions in a drug that has just been started to use in the world in breast cancer. So I think it just has to do with the pharmacovigilance of the prescribers, because they follow these. I think they follow these patients closely. For example, with the new cancer drugs that are appearing all the time, they follow them very closely, and for example, they are giving the drugs in cycles, and they follow the liver tests before each cycle. So I think we have to rely on these doctors as well, not only.
So Ari, on the cancer drug question, because there's actually in our report, there's a large section on cancer drugs that are very detailed tables, both of old chemotherapy agents as well as new drugs, and we have a litany of information about that. But I wonder whether you might comment from where you sit, Ari, about the monitoring for hepatotoxicity in patients with cancer on various anti-cancer modalities. Should we monitor patients all the time? And what should we do with the information that we get while we monitor patients, since liver test abnormalities are quite frequent?
Well, thanks, Mark. This is, I think, one of the biggest challenges in drug development, and I think in post-marketing it becomes even worse, even more challenging, because patients with cancer, especially advanced metastatic cancer, have so many potential causes of abnormal liver tests that just following and reporting elevated liver enzymes is really just a part of the assignment that we have. The most important part is figuring out if those abnormalities are actually related to the drug, and that is not always feasible in post-marketing, because it requires a very elaborate evaluation for other potential causes that patients with cancer would almost invariably have. I mean, is it an obstruction of common bile duct? Is it metastatic disease progressing rapidly? Is it a systemic infection? Is there a portal vein thrombosis? Hepatic vein thrombosis. So I think I think monitoring is important and is done, but I think the data that we receive many times, especially post-marketing, is very complicated to understand, because it's not always clearly evaluated to know if it's related to the drug or not. So I think what Einar just mentioned, is when they have very specific patients that they were reported, and they have the ability to evaluate very thoroughly if the cause was the drug or not, I think that is a very valuable part of the post-marketing monitoring work, to be able to really understand that this is related to the drug and what is the spectrum of the drug toxicity.
Right, and I agree with you. I think one of the great challenges going forward with chemotherapy and combinatorial therapy is how can we disassemble the contributions of each component in a therapy with accuracy. And we need more than pharmacovigilance. We need some kind of organized system to collect data to be able to, over time, determine what each contribution is, and whether those are registries and expert systems to follow patients is really a very important challenge, and one that we haven't fully addressed. Now I want to go to Raul. We had a really, I thought, perceptive question about the R value. And the question was that when you initially look at some individuals who have liver injury, they start with a hepatocellular pattern where the R value is greater than 5. But quickly, as they evolve their injury, it can go into a more cholestatic picture. And the question is not so much what's the convention of when do you actually measure the snapshot, but how should we think about the idea that some patients seem to have more of a kind of mixed effect where they evolve their injury over time. And Raul, you looked at this question of modifying the R value. And could you just talk a little bit about this question, because it's an important one also with reference to prognosis when there's a Hy's Law case. Raul?
Yeah, yes, I think it's a difficult to answer question because of consensus and practice guidelines recommends to establish the R value at the very beginning of DILI onset, because of we really know that the pattern of liver damage may evolve over the time to a mix from hepatocellular to a mix of liver cholestatic pattern. So we need to agree upon when is the best time to define the pattern of liver damage. And different studies have demonstrated that hepatocellular pattern may have a worse prognosis, patient fulfilling Hy's Law than mixed or cholestatic one. So probably it's important to take this into account, that the prognosis is different. So when if you decide to define the pattern later on, you may overlook this worse prognosis, for example, for the hepatocellular type, if the pattern is already mixed. So it's, I think it's a difficult to answer question, but I think at the very beginning of liver damage, I think there is a consensus about the fact that it's the better time to define the.
And just to add to that, one of your studies from your group and also from Dylan showed that some individuals who have a high R value, but also have high alkaline phosphatases above just very low levels, actually can be bad actors. So that sort of suggests that there are patients who have a kind of more kind of muddled, mixed kind of, or evolve into a more cholestatic picture as they evolve, are not always safe from a bad outcome, right?
Yes, you're right. But you know, the thing is, when in our studies, when we try to establish prognosis with analytical variables, we use the variables at the DILI recognition, not later on. So, for example, for the High's law, the new High's law from our group, we show that alkaline phosphatase may be above 2 to false, and this not diminish the risk. This really confers a similar risk that patient who had the alkaline phosphatase below this threshold.
Right,
So,
and I think, and just again, I think it's quite interesting that there's some of the histopathologic studies where biopsies are done sometimes don't coincide exactly with the R value either, especially in aging patients. So I think this is an interesting question. I want to just move on to causality. There was a question about causality, and very correctly pointed out by the questioner that DILI, which is a 4-letter word, really implies a causal association with the drug. And Ari, I want you to again really emphasize and talk about this question of the probability of causal association as part of what we call DILI.
Yeah, absolutely. I agree with the premise of the question that when we say DILI, we already mean that we went through a causal evaluation process, and now we are assuming that this liver injury is related to the drug, and that I think is a potential confusion. So I can't overemphasize the importance of that process when we detect a liver injury, of assessing whether it comes from that particular drug or not. If it doesn't, then we cannot use the terminology of DILI shouldn't be used if we're not sure this is related to the drug.
So one of the things that comes up is RUCAM, which is an algorithmic causal association test. And somebody actually asked us in advance of the meeting about automated algorithms such as PV-RUCAM or eRUCAM, and where we're at with that. Does anybody want to sort of jump in and sort of tell us about this work in progress with reference to algorithms? Einar, do you have any views on this?
Yes, actually there is an abstract that has been submitted to the AASLD meeting that has shows work that has been done with the new RUCAM, or it's called RECAM, reanalyzing this causality assessment. And E is also for the electronic. So this shows that by analysis of the cases from the Spanish registry and from the DILI, that this new tool has a better reliability than the older instrument. And maybe Raul can also, because he has done most of the work with the patient for the Spanish registry, might comment on that.
Raul, do you have a couple of comments on these various kinds of RUCAM? I know you've been working with the some of the Dylan people on this. Where are you at with that?
Yes, Yes, but you know, the results are still in Astra 4. I think we need to discuss in more detail the results, but you know, I agree with Einar. It seems to be a more objective method and has it's more evidence-based. This is important because the old RUCAN was not really evidence-based, and this is based on the data from these two registries. And I think it's a refined method, really, more objective and intended to be used in a computer ways. Base for, you know, so I think it's going to be an important addition for causality assessment.
So obviously, as we get better at this forecasting of causality and so on, we'll need to have more information and knowledge about exact mechanisms of injury and different kinds of patterns of injury. And it's complicated because, as I'll point out in a moment, DILI is not one kind of injury. There's actually a collection of different injuries, which we call DILI. So a RUCAM algorithm has to kind of include all of them, and that's sort of what the challenge is in some respects. Eventually, we'll get good at it, but I think everybody on this call agrees that it's a work in progress. We got a call here. We got a question here about software modeling and some of the simulation tools that Ari touched on: transporter interaction, ROS generation, mitochondrial toxicity, and so on. And we often, as part of the workup of a drug, often in the IND phase at FDA, when there's a question mark about hepatotoxicity, we'll get information about some of the effects of a drug in simulated populations on various steps in handling clearance by hepatocytes. Again, this is a work in progress, as Ari has said, of being able to absolutely predict based on findings what you're going to get in clinical trials. But I think we should also tip our hat a little bit to this kind of work. And I guess Ari might be, since he spoke about it, you might want to just sort of say a little more to it than just say that at this point it's still, it still has some more potential, hurdles to overcome in some cases. Do you want to talk a little bit about those simulations?
Yeah, you're referring to the preclinical assessment of the risk of DILI, and this is still, as you said, it is not only a work in progress, it is extremely controversial, and there are different approaches in different companies, and it goes all the way from some drug developers that would cancel the development of a drug when they find one of those predictors to be active. So if they find that there is a reactive metabolite, they will just discontinue the development. And it goes all the way in the spectrum to companies who will never use any of those to discontinue a development of a drug. They will use it to guide the future development and to try to monitor closer and to try to assess better during clinical development. But so this is a very controversial area. It is still developing. I think we still need a lot of data to try to assess the validity of those and the strength of those predictors.
Let me sort of sound an optimistic note, though. We actually look at a lot of this kind of data at FDA. Again, in the space a little bit of kind of like not absolutely validated prediction, but we learn about a drug from this kind and its potential effects from this kind of work. And I have to say, from various projects that I've seen, it's been at times very insightful and very useful to have this kind of information, because it gives us context when we do see a liver signal, and allows us to focus on the translation from these in vitro effects and the simulation modeling effects to what we're looking for clinically. So in the medium to longer term, I'm actually very optimistic that these tools are going to have a very important role in drug development. And I think we're a little bit like the weather forecasters were in the Second World War, we don't have yet all the predictions in place, but we have some of them. So I think our answer would be something as a work in progress, but important work. I think that I want to go back to the problem. We've had a number of questions about herbal products, and I want Hervé to talk a little bit from where he sits about what his view is about the herbal product and the problem of going from a kind of complex mixtures of plant products to regulating a hepatotoxicity question, since it's a growing area of concern. Hervé?
Yes, thank you, Mark. Thank you for the question, which is, in my opinion, about difficult.
It's a very difficult question, yes.
So I'm sure that Harry will answer to me. And Raoul, no, we try to do something because it would be so long to explain more. We can't answer this question immediately. I discussed with a lot of people from biochemistry to finally pharmacovigilance and another one. We are trying to initiating an initiative with the Chinese government, the IUFAR and the ISOP and the CIOMS, of course, to try to move on. But honestly, I have nothing concrete to see. We really, and many other people understood that it was a big issue, and especially in the US, if you take Victor Navarro in Philadelphia, which was a lot of in this way. But to be honest, we must now create a new way to approach this problem. So, Mark, I know that you know that more than other people.
Right. I think that it is the hardest question to answer. Yeah, go. Sorry, who's next? Who wanted? So I think we're coming close to the end of our session, yes. But I was asked to give a 5-minute or so summary by the meeting organizers about some of the key points in this landmark report that today we heard about from our excellent speakers and panel. So just to kind of give a brief summary, to begin with, a thorough analysis of DILI liability surrounding a new drug or biological agent, both during its development with the tools that are available to us and that are preclinical and then in clinical trials, and even later in a post-market setting. This is a key point, is a life cycle process. It's an iterative approach to evaluate new data over the entire life cycle of a particular drug. It gives us an opportunity to periodically calibrate associated risk for DILI. And it allows us to introduce regulatory actions when needed, with effective communications and label modifications, as well as to optimally manage patients who may receive the drug in clinical practice. So as I already kind of said before, DILI really is a 4-letter word, but in fact, as Ari pointed out, the report emphasizes that DILI represents a number of different kinds of injuries of the liver caused by different drugs and agents with different underlying mechanisms and clinical pathologic phenotypes. Some forms of DILI are direct and related to exposure levels, but others are indirect, caused by unintended yet known biological effects of the drug. And then many forms of DILI are idiosyncratic and rare, but can be life-threatening in susceptible individuals. So again, when we review clinical trial data, we become especially concerned if we identify even 1 or 2 acute hepatocellular injury cases with jaundice caused by the drug. Once all alternative possible causes of liver injury have been systematically excluded, we would call them Hy's Law cases, and they portend that the new drug will have a liability for causing acute liver failure, rarely but unpredictably in similarly treated patients in the post-market. So we need to also recognize that other phenotypes of DILI, such as cholestatic hepatitis and a number of different subacute or persistent forms of liver injury, can be troublesome, even life-threatening in certain clinical settings and vulnerable patient populations. And we heard about this from Ari, about the different diseases, underlying disease of concern. There's been a whole litany of recent publications on these diseases in DILI, and the listeners can go into look into those. So the CIOMS group members all would agree that when we model DILI to make predictions about the associated hepatotoxic risk for, let's say, a new anti-cancer drug or an anti-TB drug, we have to take into account the injury phenotype in addition to the severity of the liver adverse event and its causal association with the drug. To assess clinical trial data, we often utilize a structured opinion method with clinical experts, since this approach has flexibility in bringing these pieces together. However, we do recognize that more work is needed to develop refined algorithms building on the current version of RUCAM that will in the future align with all the different phenotypes and clinical signatures of DILI that we now recognize for providers to apply in their clinical practice. And then finally, as we just heard from Hervé and from Raul, in the big picture, there is a rising worldwide usage of herbal products and dietary supplements. Some of these have a clear liability for hepatotoxicity, and how we should evaluate their DILI risk with the scientific and regulatory tools that we currently have at our disposal is an enormously important challenge. It requires a multidisciplinary approach, everything from bringing to bear innovative tools that accurately measure their chemical contents and toxicologic attributes, to those that elucidate key epidemiologic factors that explain increases in their usage in so many different demographic groups and communities across the globe. All of my work colleagues in this working group would agree that the 2020 report takes a deep dive into many of these issues, and although it's not an official document issued by any regulatory authority per se, it's a state-of-the-art consensus document that catalogs what we've learned so far, and it also looks to the future for groundbreaking advances by our industry and academic partners. So in closing, we want to keep all of you as key stakeholders in this arena engaged. One venue that has gained traction in our community is the meeting on DILI that's co-sponsored by the US FDA and the American Association of Liver Disease that is held every 2 years. And we're working to finalize plans for the next cycle of this meeting, DILI 2021, which because of uncertainty surrounding COVID-19 would inevitably be a virtual meeting. But if we get it approved, it's likely to be held in the 3rd or 4th week of April 2021. So please stay tuned. And with that, I'm going to hand the baton back to our organizers of the meeting.
Thank you, Professor Mao. We would like to give a special thanks to the participants and the Center of Regulatory Excellence for organizing and facilitating this webinar. Thank you and have a nice day.